The first DNA isolation was described by the Swiss physician Friedrich Miesser in 1869. This was done by identifying an acid-soluble phosphorus and nitrogen-containing substance which he called nuclein in the nucleus of leukocytes, obtained from the pus on surgical bandages. For the isolation of this new substance, in 1889 it was renamed into nucleic acid by his student Richard Altman. Then, Miesser used a successive solvent and alkaline extraction method with subsequent acidification. In 1944, 50 years later, the better-known story of DNA started with Avery, MacLeod and McCarty showing that the hereditary substance is in fact nucleic acids and not protein as assumed in earlier studies. Followed by the observations of Erwin Chargaff, in 1953, James Watson and Francis Crick finally discovered the molecular double helical structure of DNA created by the base pairing of adenine with thymine and cytosine with guanine. The blood contains two different blood cells. In first place, the red blood cells contain lipids, carbohydrates, minerals, hemoglobin and iron. Secondly, the white blood cells are carriers of proteins. For DNA extraction purposes, it is vital to isolate the white blood cells in order to get a pure DNA. The centrifugation is a separation method in which the rotational speed around an axis increases the force of gravity. Thus, the faster you rotate, the greater the gravitational force will be and vice versa. This force generates a sedimentation of the components of a specific substance and this separation is based on the size and density, where larger and denser particles are precipitated. At higher speeds, smaller components are sedimented. The proteins of the blood remain in the supernatant liquid and require more centrifugal force and time to precipitate. Thus, higher evolutions are required along with an extended time to precipitate proteins from the blood and thus extract pure DNA. The aim of this practice was to isolate DNA from human blood. To do this, the blood was washed with saline to remove the blood plasma and then the cells were broken, using a detergent, to release its nucleus. Proteins were then removed from the cell membranes using an enzyme that degrades them. In order to make the proteins heavier and precipitate them by centrifugation, a salt was used. Finally, the DNA was precipitated by adding an alcohol. At this point of the procedure a species of white speck was observed. The result was a complete disclosure and extraction of DNA of the human blood sample from the nurse of Gymnasio Campestre. The test tube evidences the double-stranded molecule as a tiny white speck. The results obtained were coherent to what was expected. A pure DNA with a considered amount could be achieved by following all the steps of the experiment. Also, this shows that in the white blood cells there is an appreciable volume of proteins that encode amino acids in order to produce genes important for the function and structure of the human body. This result suggests that the use of different elements in a determined pattern are useful for an accurate extraction of DNA of a specific human blood sample. It reveals the relevant use of saline as a solution to wash cells, the detergent as an element for the outbreaks of cells, the enzyme as an effective component for the degradation of proteins, the salt and the centrifugation as a fundamental for the precipitation of the materials of blood, the temperature as a catalyst to accelerate the reaction between two solutions, and the absolute alcohol as a useful tool for the complete exposure of DNA. So that, the function of each element in a rigorous procedure allowed a pure DNA extraction.